Okay, so what this particular chapter is going to be about is on enzymes and how this is going to be different from students doing the pure biology in O level. Is the only difference is you have to understand the role of enzymes and also the activation energy. Whereas students taking the science biology in O level and N level, they would have to actually know the how the factors and everything else, and not so much on the emphasis of the activation energy. However, properties of enzymes, a lot of key hypotheses, and the factors which are your temperature and pH in influencing how the enzyme reaction takes place is important. Okay, so if we take a look at the particular learning outcome, you will need to actually know how to define an enzyme, and this can be an example of an essay question where you can say, state the role of enzyme, or it can even be what is an enzyme, and it'll be 2 to 3 marks. So what they want is actually to first define what is really an enzyme. In order to understand that, you need to know that enzymes are catalysts. So what is a catalyst? A catalyst is basically a particular substance that helps to alter the speed of the reaction. So altering the speed, right, means it can actually go faster or slower, but in the case of enzyme, most of the time it actually speeds up a chemical reaction. What are some examples of this chemical reaction? We can say mostly your digestion reaction, or mainly those involved in digestion. Like when there is a particular um, substrate, we call it the substrate, being broken down into its product. And of course, you will use the word enzymes to actually annotate it. So it's something like a chemical word equation. Actually, okay. There's no such word as chemical word equation. You just treat it as a word equation where the substrates are basically, and the substrates, the name of the substrate and the product is mainly the name of the nutrients. Okay, next, and what's so special about catalysts is that at the end of the reaction, they do not actually alter any. They do not chemically. They are not chemically changed. Okay. Now what? So are we able to visualize this? Is maybe the shape. So based on the shape of the enzyme, right, it doesn't really change so much, such that it can alter its property of catalyzing the reaction. Okay, next we look at then what is then enzyme? How is it related? So while we understand that catalysis, number one, okay, must actually speed up the reaction. The second one is that at the end of the reaction, they remain unchanged or remain chemically unchanged at the end of the reaction. So imagine catalyst as a very big box, and within a box, when you open up the box, you see that there's all these small little toys inside. Enzymes are an example of these toys where it's a subset of catalyst. So enzyme being a subset of catalyst, right, it must have other unique properties that set it apart from catalyst. So in that sense, so an enzyme, right, is actually a protein. So if you remember, we actually talk about the different nutrients group. So protein is an example of the enzyme is an example of the protein nutrients group. And what this does is it actually functions as a biological catalyst. When we see biological, it means something related to living organisms, body and things like this. In this case, we are talking about human. And the thing is, if you look at point number 3 and number 4, these are what are the properties of a catalyst. So you can see that an enzyme, right, is actually a protein that functions as a biological catalyst that helps to alter the speeds of chemical reaction and remain chemically unchanged at the end of the reaction. So these 4 points, how to phrase it into one statement if they ever ask you to define what is an enzyme. An enzyme is a protein that functions as a biological catalyst, full stop. It alters or speed up chemical reaction and remain chemically unchanged at the end of the reaction. So you can see that most probably the first 2 points will give you 1 mark in exam, and last and the point number 3 and point number 4 is the second mark in exam. Uh, for the definition of enzymes. Okay, if we then let's move on further. So how is enzymes able to function that well? Okay, or how does generally enzymes react? So think of it this way: activation energy. So if we have a particular normal reaction, we realize that any reaction has some form of activation energy to occur. Now, what is this activation energy? Activation energy is the amount of energy required to start the reaction. So in this case, right, let's say we look at this diagram here, you will see that activation energy is this period over here, where where it's the initial start. So you can see that at the start of the reaction, right, there is this basic hum, and this hum is the one that is the amount the amount of energy that it needs to start a reaction to move forward. So think of it, so it's like a barrier. If you do not meet the energy, let's say you are starting a stopwatch and you only provide this amount of energy until here only. Your reaction will not proceed forward because you are missing this extra amount of energy for it to overcome and for it to start the reaction. So there's a lot of difficulty. So then how then enzymes will actually function is it helps to bring down the starting energy even lower. So you can see at initial start of the activation energy without the enzyme, it's actually quite high. The hum is very high, the height is very high. But with the role of the enzyme, the enzyme would actually reduce the amount of energy to a shorter period here. And this will then allow the same amount of heat energy to overcome the reaction and to start the reaction, to kickstart. Now, why do we need this activation energy? This activation energy helps to safeguard reactions from happening spontaneously. So, for example, in your human body, there is a lot of reactions happening in your human body, but you don't see you like suddenly combusting into thin air or suddenly your hand burns. The reason is because the activation for certain reactions in your body has a very high activation energy. So even at a very hot temperature, even if I increase the temperature as a supply of heat, as a supply of energy, you don't suddenly see yourself burning. However, if let's say it's too hot to the point whereby you will start to undergo decomposition, okay, like if you let's say you throw a petrol bomb onto somewhere and then you catch fire. Now your body will start to actually burn even more when this fire supplied so much heat energy that it's above the threshold of what your body can take. So the activation energy is being overcome. Therefore, your body will start to burn, start to undergo decomposition, and that's the main purpose of the activation energy. So the role of enzyme here is not to change the reaction, like from in this case to change to actually convert glucose, oxygen into carbon dioxide and water, um, in the form of changing its molecular structure. But the enzyme is there to help to reduce the activation energy such that it is much easier to start the reaction. And by making the reaction to start more easily, you indirectly actually helps to reduce the time needed to start the reaction. Hence, it comes up with its property of speeding up the chemical reaction. And of course, throughout this whole thing, the enzyme doesn't actually become chemically changed. It remains chemically unchanged throughout the whole reaction. And because of it having this unique property of remaining chemically unchanged during the whole reaction, it can be used in small quantity and can be reused again and again. So what are some reactions that enzymes catalyze? Digestion is one big thing that we'll be talking more in the next chapter on nutrition in human. Where digestion is whereby, if you think about it, there are food molecules when you eat. Let's say you eat rice. Okay, these molecules are generally large and they are insoluble in water. So what happened is the molecules, and when you actually take it in, they are just too large to pass through the partially permeable membrane of the cell. If it's not able to pass through the partially permeable membrane of the cell, your cell who's who has organelles, right, cannot use all these molecules to actually build other things. And in that case, right, whatever you eat doesn't it's not fully utilized. It will be like maybe removed out from your body when you go to the washroom and things like this. So what we want to do in digestion is to break down these food molecules into smaller soluble substances such that when it is small enough, when the molecules are actually small enough, they are able to actually pass through the partially permeable membrane. Otherwise, whatever you eat will not be used properly. That's how protein powder generally helps to build muscle. Where if you take a look at the nutritional fact, right, you will see that some of these amino acids they're actually grinded in at the molecular level to be soluble. So it helps to speed up your muscle recovery as compared to maybe you take chicken breast meat, which your body will then need enzymes to help to break it down even smaller so that your body is able to act on all these that can utilize the amino acids from the chicken breast to build your muscle. Okay, so this is one example of digestion. Then what are some of the names of the enzymes that are involved in digestion? They are like your amylase. The amylase tends to digest starch to maltose. This can be written in the form of a word equation where on the left hand side of the arrow you will write the name of the substrate, the starch, and the end of it the product, which is the maltose. So the enzyme that is involved is what we call the amylase. So similarly, maltase is the one, and like the trick to look at it is most enzymes end off with ASE. Okay, and of course the naming of the enzyme usually follows the name of the product that is going to be formed. Like in this case, when you use it will actually follow the name of the substrate. So in the case like maltose being digested into glucose, okay, the action of maltase is the enzyme where maltase through the nail of the malt follows the substrate, and the second part, which is the ASE, indicates that it's an enzyme. And in the first 2 points, you will see that these two enzymes are responsible to digest carbohydrates. Protease helps to digest proteins. And finally, lipase helps to digest fats. Of course, protease is a very generic name. There can be multiple enzymes, specific name that falls under protease. But these are some of the few examples that you will need to know. So next, we take a look at reactions that enzymes catalyze. So there is two types of reaction. There's two types of reaction in general in your body. Reactions that helps to build cells or to build substances in your body. That's one. And substances that are being and reactions that help to break down substances in your body. So there is a build. There is also a breakdown. And together, right, this will actually gives you a overall process like body how much your body can take. So when we look at this, right, we usually use the term metabolism to understand the overall is your body actually building enough cell? Are you gaining weight or are you losing weight more? So it's actually an overall net movement. And the building of substances, right, we call it anabolism or anabolic reaction. And the breakdown we usually call it catabolic. So if let's say, right, you have a higher anabolic rate as compared to a catabolic rate, so your anabolic reaction is higher as compared to your catabolic reaction, you'll realize that you are actually going to maybe grow higher in mass. So your metabolic rate is positive. So positive metabolic rate. And in this case, what you mean is maybe you will increase in mass. This is one example. However, if your body, right, has a lower anabolic rate as compared to catabolic, that means you are actually breaking down more things than building more things in your body, you would have a negative lower metabolic rate. And in this case, maybe your mass will reduce. So let's say you are giving you the example of maybe you're dieting. Okay. So when you diet, right, you are actually taking in lesser substances. And when you're taking lesser substances, you in a way you don't really build things. You don't build fats and in your body and things like this. But then you break down more things because of respiration, and so you utilize whatever fats is in your body and you slowly break it down. So in that sense, right, you will have a rate whereby you have a higher rate of breakdown as compared to building up. And overall, this will lead to you having a higher metabolic rate and therefore resulting in a reduction of mass. So that is an example of metabolism. Okay, so next we look at the examples. So anabolic reactions are the building up of complex substances, like building up of protein from amino acid, and catabolic reactions are basically the breakdown of substances. But of course, just now the example I gave you can be quite confusing because um, anabolic reactions, catabolic reaction has to be placed with one specific reaction to discuss. Then it's much easier to understand. But overall, this is what the general idea is. Do not worry. This portion is not really tested that much. It's more towards of you understanding the purpose or what are some processes that enzymes actually use. So in this case, like what I've shared, as enzymes are usually named after the word ASE. Yeah? So as long as you have the word, as long as you identify the substrate, the substrate, you just add the ASE behind, that's the name of the enzyme. But this is not the 100% proof. This is just going to occur most of the time. Okay, so sometimes you will still need to remember, but generally for O level and N level, they tend to use ASE as a form of indicator that it's actually an enzyme. So in exam, especially in MCQ, if you ever come across like, oh, which of the following are enzymes, you will naturally know which are the ones. And enzymes, because they are protein in nature, what will happen is they will actually give a positive test in your burette test. That means you actually follow a violet coloration. So another exam question that they can come out is which of the following substance can actually result in a violet coloration in burette, and then you got A, maybe a carbohydrate, B, you have a fat, then C, a name of an enzyme, and D, maybe something else. So water. So in that sense, right, you don't really get to see the word protein per se in MCQ, but and the way to trick you is they want you to recognize that enzymes are not carbohydrates. So like let's say I use maltose as A and then B I say maltase. So the answer will be maltase. Because maltase is an enzyme and enzymes are protein and therefore they are able to show a positive test using the burette solution test. So how are enzymes classified? They are classified according to the chemical reactions that they catalyze. So are they undergoing So if we look at this, this is hydrolase. Hydrolase are basically reactions that requires water to break it down into smaller substance. So this one, like I say, not so important, but generally that's how that's what the type of reaction they usually catalyze. Where water is definitely a very important solvent to help to cause any form of reaction. Okay, next we look at what are some of the modes of enzyme action. So in this case, it's mainly describing the log and key hypothesis and also to understand the temperature and pH in terms of how it affects the rate of an enzymatic reaction. So this is important. If Please memorize this, please remember this, as this can be a very good example of an answer to an essay question. So if let's say they will ask you what describe the role of an enzyme, this is the answers that they are looking for. Okay, so it's different from basically defining an enzyme. So do take note. So in that sense is like enzymes, they help to speed up the chemical reaction. In if let's say you say it alters the speed of reaction, you won't be getting the mark because altering the speed of reaction is a very generic sequence. There is no direction. You can either slow down or speed up. And most of the time for enzymes, they generally speed up the chemical reaction. So what you need to do is when you write your answer, you have to say like enzymes alter the speed of reaction by speeding up the chemical reaction. Okay, so the keyword is speeding up. Next is enzymes are required in minute amount. So minute amount means small amount. Now why is it how is it able to fulfill this requirement is that reason being is because the enzymes at the end of the reaction, right, they remain chemically unchanged. And when enzymes they remain chemically unchanged, you can reuse the enzyme again and again and again. Therefore, the enzymes, right, because you can use it again and again and again, they you only need a very small amount. And this is very important, even though this one statement it may mean nothing, but if let's say you are in a industry that deals with enzymes for to manufacture something, what you want to do is to save costs. So if let's say right you have to buy materials such as an enzyme to keep on to refill, to reuse, to perform the reaction, and you need large quantity of it, you are actually bleeding a lot of money. So so one unique property of this and of enzymes is that you don't need a lot. You can save costs by reusing. By reusing the enzymes in your reaction, right? The only money you have to spend is the substrate, and that's all. And maybe the machinery and things like this. But then the enzyme, because you can repeatedly use it for a longer period of time and of course in very small quantity, you can actually save a lot of money. But also that's also being determined by how fast how fast you can speed the reaction, because by using even more enzyme, your reaction can actually proceed faster. So it's a matter of finding the right balance of the right concentration such that you get the optimum speed of the reaction that gives you the maximum output. That means the maximum volume of the product form. So that's an example. Okay, next we look at enzymes that are highly specific in nature. Now, what we meant by highly specific in nature, that means they're quite choosy. Okay, so for example, that each chemical reaction inside a cell is only catalyzed by one unique enzyme. That means, right, if I'm enzyme A, I will only um, catalyze reaction A. I will not catalyze reaction B, C, or D. Okay, one example is, let's say I'm a boy. I will only go to the male's toilet. I will not go to the female toilet. So this is very specific. So this is very specific. Okay, and then an enzyme is specific due to its three-dimensional shape. So it's how the whole 3D shape of the enzyme, right? Especially the active site that helps to determine or to decide whether this particular enzyme belongs to which specific chemical reaction. And the active site, right? The shape of the active site generally follows the shape of the substrate. So when we look at enzyme specificity, one very good demonstration or experiment is I would say experiment, but a theory, a hypothesis or theory helps to describe this very clearly, and that's the lock and key hypothesis, which explain the enzyme specificity. So and of course this dependent on the active site. So what's the active site? Just imagine it the whole of the lock. So think of it this way: you have a key, okay, and you have a lock. So lock A, lock B. So the shape of the key is very unique, and of course the keyhole of the lock is also very unique. So let's say right, I have maybe the keyhole will only accept this kind of shape, whereas for the lock maybe you will accept only this kind of shape. So what will happen is the key will only open lock A and not lock B. Why? Because the shape the shape of the lock or the shape of the key is only able to fit the lock hole of A, but not the lock hole of B. And this is similar to your how the 3D structure of your substrate is, the 3D structure of your enzyme is, whereby the enzyme and the substrate, right, has different kinds of shape. For example, let's say the key, let's say the enzyme here has this shape where this is actually the active site, okay, and the substrate has this shape. And of course, you have another substrate, maybe it's a square. So which one could actually fit into the enzyme active site? Most probably is the triangular substrate. Why? Because just nice is being able to fit in nicely. Pardon the accuracy of the drawing, but this is what usually it means. Okay, so because of that, it can then create a complex together. The enzyme will then slowly catalyze the reaction, and later maybe the substrate change shape to become something like a circle, two circle, where it no longer fits the active site of the enzyme, then it will start to exit and form it. Ready, and therefore, at the end of the day, you could see that the shape of the enzyme, right, it always remains the same. Only the substrate changes its shape, and no, and through then, it will no longer be known as a substrate, it will be known as a product. But if you look at this substrate, it is not able to fit into the active site. And if it's not able to fit into the active site, it cannot catalyze, the enzyme will not be able to catalyze the reaction, and therefore it will not react at all. Okay, so that's what they meant by a lock and key hypothesis. So an enzyme has a specific three-dimensional shape that contains the active site, in this case, and from there you can see the shape, this particular shape of the substrate, right, falls in nicely onto the mouth or the active site of the enzyme. So the enzyme would have a specific 3D shape that contains the active site, and this is where we say that the shape of the active site is complementary to the shape of the substrate. So if we look at the second point here, the substrate with the 3D structure has a complementary shape to the active site, and therefore it can fit it nicely, forming an enzyme substrate complex. Now in exam, right, it is good to actually write down the name of the enzyme and the name of the substrate that is being involved, rather than you say a generic thing like, oh, only the substrate will fit into the enzyme. So please, if you can, change the word substrate, change the word enzyme to the names, to the name substrate and enzyme that is in the question exam paper. Okay, and usually if the exam they ask you use a named enzyme or substrate to explain the lock and key hypothesis, you are required to base on whatever you have memorized and remembered and to put it in. And the starting statement you have to actually say which is the lock and which is the key. This one, please read your textbook. They actually have a very good example that will tell you which is classified as a lock, which is classified as a key. Um, and do your own reading to find out more from the Marshall Cavendish textbook. Okay, next is we look at the third reaction where the chemical reaction occurs and then the substrate is then converted into the product. So you can see, right, that eventually there will be some form of reaction between the substrate and therefore it will slowly change in shape, leading to it forming a product and having a shape that no longer is complementary to the active site of the enzyme. So the so next what happens is the product then detach itself from the active site, and these enzymes will help to remain unchanged at the end of the reaction. And therefore, you see, at the end of the day, the enzyme still remains the same shape. So it can be reused again, but not the product. The product will be maybe removed from the reaction and things like such that a new substrate that is the same substrate that has the shape will start to react again. So this always occurs. Now what happened is this whole reaction will stop if, let's say, the enzyme lost its shape of the active site. And this term, very important, we call it denaturation. So please remember this term. Okay. And it's a common term that we use in exams. So in an enzyme reaction, if you don't really see the word denature in your answer, right, then most probably something is wrong. There is most probably one portion of your answer that you answer wrongly or what, unless you are very lucky that they never ask anything about denaturation. So denaturation is where the enzyme actually loses its three-dimensional shape, and this can be influenced by two factors, your pH as well as your temperature. So having lost the three-dimensional shape, then it will not be able to carry out the function. So the reaction will either remain at a slower pace or it will basically not happen at all. So a change, so in this case, do take note that there's two factors, the heat, extreme high temperatures of heat can lead to denaturation of chemicals such as acid and alkali, which helps to disrupt the shape of the active site. Now, if you want to understand more as to how the acid and alkali actually disrupt the active site, is like, just imagine this way that there are, in chemistry we learn ionic bonds. So your active sites are also helped by maybe ionic bonds or covalent bonds. And acid and alkali helps to actually produce the ions and disrupts the charges of the bonds. And when that will when that is going to happen, and when that is going to happen, and when that is going to happen, and when that happens, right, you will see that the bond starts to weaken and be disrupted. The bonds that helps to hold onto the shape of the active site. When these bonds are actually being disrupted, then the whole reaction would actually um, your whole active site will actually be destroyed, and that's where your enzyme has undergone denaturation. Do take note, enzymes do not die. So do not write things like extreme high temperature enzymes die. This is a wrong thing to do. Instead, enzymes they will actually undergo denaturation. So when an enzyme denature, there is this loss of the active site. And due to this loss of active site, the substrate will no longer fit into the enzyme's active site, and therefore there is no reaction. So this is how it looks like. Do remember the enzymes are still like three-dimensional in structure, and while substrate also is 3D in structure. So it's not very like 2D in this way, but 2D helps to understand something. Okay. Next is enzymes are affected by temperature. So I will most probably create another video to go in deeper understanding as to how to read graphs like this in enzymatic reaction to help you. But in this case, I'm just basically explaining generally how enzymes can be influenced by temperature and also pH. So enzymes are very inactive at very low temperature. This is the reason of this is that you see, in order to form the enzyme substrate complex, your substrate must be able to collide with the enzyme. Without that form of collision, that means meeting each other or hitting each other, you will not form the enzyme substrate complex, and therefore you cannot have any form of products formed. So what actually happened is that at very low temperature, you have very little supply of kinetic energy. What is kinetic? Kinetic is something like moving energy, or energy that requires for you to move. So to make you move. So at low temperature, you have low kinetic energy. So all your molecules are actually moving very slowly. And when it's moving very slowly, the frequency of it colliding with each other, a substrate and an enzyme, is very low. And hence, this will actually decrease the chance of the substrate, right, to have an effective collision with the enzyme. And therefore, it will not have a reaction. So this actually happens at the start at A. And of course, by increasing the temperature, the rate will actually increase because you are supplying more kinetic energy. and therefore increasing the chance of the enzyme being able to collide with the substrate. Next, as there is an increase, like what I say, when there's an increase in kinetic energy of the substrate, not of the substrate, of the molecules, there's an increased chance of collision. So in the sense, right, your frequency of collision increase. And when your frequency of collision increase, your frequency of effective collision also increase. And this will then increase the rate of formation of the enzyme substrate complex. Now, there is then this particular nice temperature whereby no matter how whereby this is the maximum amount of heat energy that you can increase such that this is the fastest rate of collision. Okay, and anything more than that, right, will result in a decrease in the rate of reaction, and this is what we call by the optimum temperature. So, the optimum temperature is the point where the rate of the reaction is actually the highest, and this is where the enzymes are most active. Okay, do take note, we use the word optimum here. Okay, and generally, enzymes have different optimum temperature. It really varies on what kind of temperature, what kind of enzyme. So, like for example, in a volcanic eruption, there might be some bacteria present in volcanoes. Most probably, the enzymes within those bacteria are pretty high, to maybe a few thousand degrees Celsius, as compared to maybe the enzymes in human, where most of our enzymes are working at somewhere about 40 to 45, or maybe even lesser, at 37 onwards. That's why right, for our body temperature, generally we try to hover around 37 degrees Celsius, because that's where most of our body enzymes are able to do. And whenever we have a fever, maybe at 38 or 39 degrees Celsius, this is where the enzymes will start to denature. And that's why and one consequence of that is you will just feel very feverish, you'll feel very weak. So that's about all for this particular portion. So moving forward, right, you can see that beyond the optimum temperature is where you start to see a decrease. You start to see a decrease in the rate of reaction. It starts to move downwards. And when the activity actually decreases, this is because denaturation has occurred due to the high temperature. The high temperature has broken the bonds with the enzyme and hence changed its 3D structure. And the active site of the enzymes will start to lose its original shape, and this is where we say that the enzyme is denatured. Okay. So you can see, right, there is four main phases in a particular enzyme reaction, which in exam you might be asked. The first one is at low temperature. The second one is at rising temperature. The third one is at optimum temperature. And last but not least, the fourth one we are talking about at beyond the optimum temperature, which leads to denaturation. So at low temperature, we say that low reaction due to the low amount of kinetic energy. Rising temperature, you increase kinetic energy, therefore faster speed of reaction. Optimum temperature is the fastest rate of the enzymatic reaction, and beyond the optimum temperature, the enzyme will have undergone denaturation. So usually in exam questions and structure questions, they tend to classify or categorize their, like, the part A, B, C, D, right, for in phases like this. So do know how to recognize it. Okay. So if I were to break down, it's like maybe A is here, so it's number one. Somewhere until here is number two. Exactly down here is number three, and the segment at D over here is number four. So that's how you usually interpret. And most enzymatic graph, do take note, it doesn't follow a bell shape. A bell shape means, right, it's actually a very even curve. So what you can see is this is actually a very gradual increase and then suddenly a very sharp downwards. So this is one property of enzyme graph that you can also take note, especially in MCQ where you're required to identify the shape of the enzyme. And do take note of the axis as well. These are important. Finally, we look at the enzymes are actually affected by pH. So different enzymes will have different optimum pH at which they are most active. So what happens is this is what we call a bell-shaped curve. A bell-shaped curve. Okay, in which right there is only one optimum pH, and it's not because it is at 7 that the optimal pH for most enzymes is at 7. No, this can vary. So let's say I have a enzymatic graph. That's like that. So I have pH, maybe rate, and these are pH. So I have one 3 here and one maybe 10 here. So what you can see is like the purple color graph, right, would actually show that the the enzyme for that is responsible for the purple color graph, which is A, okay, will have an optimal pH of 3. That means this A is very active at an acidic medium, whereas B is actually very um, active at an alkaline medium. So if at for A, it will start to denature at extreme pH where it's moving towards the alkaline region or even more acidic region below 3. Whereas for B, right, we are seeing that it has an optimum pH of 10, so anything beyond 10, it will denature, or anything below 10, it will denature. So that's an example of how we actually see the pH being influencing the enzyme. And so you don't really need to explain how pH actually denatures the enzyme. All you need to know is how to interpret it. So most of the time, the focus should be on temperature. And there are also other factors that actually influence enzyme reaction. You can have the concentration of enzyme. You can have the concentration of enzyme. You can have also the maybe the concentration of substrate. So all these kind of examples, right? I will be creating a separate video to actually explain all this. But as of now, this is the main basic things that you have to know. Okay, so in a follow-up question, they will always say like, oh, give one factor that there is other that is being discussed that actually affect the rate of reaction for enzymes. There's only two factors that influence rate of enzyme for students doing the science bio N level and O level. They are your pH, they are your temperature. Okay, so it's either one. So that will generally maybe give you 1 mark. Okay, so that's the end of this particular chapter. Of course, do remember to like, subscribe my channel, and click the notification button to click the notification button to actually help to share this so as to get more notification of new videos. Okay, thank you. That's all for today.